In the previous video, we have understood that there is a cell-cell transfer occurred between macrophages and pancreatic cancer cells in a way that the different pancreatic cells behave differently based on what are the informations transferred from the macrophages to the pancreatic cancer cells. That is, some cells have more migrating characteristics, whereas some cells has more tumorigenic characteristics. So in this video, we will understand in detail what are the metabolic reprogramming occurs in those mutated cells to understand why they behave in a different way. So what the author does is that they do the RNA transcriptomes analysis of this RNA transcriptome analysis of this different W fluorescence cells that they have studied in the previous experiment, and then compare them with the wild type cells, wild type pancreatic cancer cells, and these macrophages cells. And what they have find out is that these cells behave more predominantly similar to the pancreatic cancer cell lines with some changes. So these are the results that has shown for SP0926. And in this case, what they find out, some of the based on this GACA, that is gene set enrichment analysis, they find out that the pathways that are mostly upregulated are this hypoxia pathway, and they have this epithelial to mesomercous transition, the hallmark of UV and glycosylations pathways. These are mostly upregulated pathways. This is the reason why these cells behave like a migrating cell lines. And two pathways are shown, GSEA of two pathways are shown here as a representations, where we can see that hypoxia conditions are clearly enriched, and also these epithelial to mesomercous transitions are also enriched. And in the panel B, they did the similar RNA transcriptome analysis and compared them with the pancreatic cancer cell lines for this SP1030 cells. And in this case, they find out that the stress-related pathways such as cholesterol homeostasis and estrogen response get disrupted, and other this in this case also epithelial to mesomercous transitions are upregulated. However, two most upregulated pathways are this estrogen response pathways and the cholesterol homeostasis. But however, the hypoxia is also upregulated, and the representation of the cholesterol homeostasis and the hallmark hypoxia are shown here. And based on this RNA transcriptome analysis, they are able to understand that when the pancreatic cells or W fluorescence cells has these migration properties, then this hypoxia EMT pathways are upregulated, mostly upregulated. Whereas when the cells have these tumorigenic properties, then in that case this cholesterol homeostasis, hypoxia conditions are mostly upregulated. After that, they did the two-dimensional electrophoresis and then find out 10 protein spots, 10 peptide spots, where they have did the proteomics analysis, proteolysis and proteomics, and where they did the proteomics analysis and find out that this 10 spot contains 16 proteins, and these 10 spots are upregulated in both SP0926 and SP1030, and based on their upregulated proteins that are identified, and these upregulation proteins, the criteria that they have used is greater than twofold upregulations. And based on these upregulated proteins, they are able to identify that two different pathways that are upregulated are this glycosylases or glycogenesis, and the other one is this cell cycle pathways. These pathways are upregulated in both the knockouts, and these things they have validated both in the normoxia condition and also in the hypoxia condition, where they have find out that the pathways that are related to this glycolysis and glycogenesis are upregulated in both in the normoxia condition, both in the case of SP0926, SP1030, and also in the case of this SP0926 and SP1030 in the hypoxia conditions. So they check the concentration of uh, pKg1 through the ELISA for this panel E in all the cases, which is related to this this glycosylases pathways. So based on that, the metabolic reprogramming, they are able to find out that when the cells behave a particular way, then what are the pathways that might be upregulated in the pancreatic cell lines. If you need to understand more about their experiments in detail, how they did the bioinformatics, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.